All right, guys. So let's get into meiosis. Okay. So this is going to be very similar to what we went over when I did it in the slideshow. Remember, mitosis, what we went over prior, is when we want to create cells that are identical to the original. Meiosis, so meiosis, that's when we are producing our gametes. So in humans, this is when we produce our sperm and our eggs. So a few terms to understand before we start. I'm going to go over all of these like I did before. And again, all of this is available in the description in your flashcards with those descriptors. So we got homologous chromosomes, gametes, haploid and diploid cells, sex chromosomes, autosomes crossing over, and the purpose of meiosis versus mitosis. Now, hopefully, based on just these videos so far, you guys understand this bottom portion right here. But I'll say it again. Meiosis, this is what we're about to talk about in this video, is the process by which we produce gametes. Gametes are those products that we make to create other organisms. So in humans, this is the process where we create the sperm and the eggs, which are the components that create another human being. Without sperm and eggs, there are no humans, okay? So this is the process that does that. Mitosis is how humans grow, okay? Mitosis is we create identical cells to the original. So the way I remember it, like I talked about in another video, mitosis is how I create my toes. Meiosis is how my parents made me, okay? So let's get into, we already talked about the purpose. Mitosis is used to make an identical cell to the original. Meiosis is used to make cells that we use for reproduction. So sperm and eggs in humans. It's a little bit different when we talk about plants and certain other organisms, but for right now, I just want you to understand what meiosis means in terms of human reproduction. So gametes, these are sex cells which have half the number of chromosomes as the original parent cell. So for example, in the average human, if everything goes correctly, you, me, most people you know, have 46 total chromosomes. 23 from your mom and 23 from your dad. Okay, that's how it is. That's how it's always been since humans were made distinct as a species. So when you produce gametes, those cells have 23 chromosomes in each of them. Because that is what we are using to pass on our genetic information. Okay, so gametes are haploid cells. Haploid, Haploid, means they have half the amount of chromosomes as the original parent cell. For example, let's say a kind of fruit. Oh, no, fruit is a bad example. I apologize. Let's say an organism has 30 total chromosomes. How many chromosomes would their gametes or haploid cells have? So they have 30 total. How many chromosomes would the haploid cells have? Have a little Dora the Explorer moment there. 15, very good, because they have half the amount of normal. Diploid cells have the normal amount, okay? So a diploid cell has the full set, okay? So we've got haploid, which is half the amount. Diploid has just the normal amount, okay? So humans have 46 chromosomes. So how many chromosomes do our gametes have? How many chromosomes are in a sperm, or how many chromosomes are in an egg? If we understand them to mean a gamete is a haploid cell with half the number of the original. 23, very good. That's why they call it 23 and me, okay? Because you get 23 from your mom, 23 from your dad, all right? This is just some more terminology. So the whole X thing is a chromosome. That's what we refer to when we are talking about this whole operation right here. When we're talking about chromosomes that are lined up together, all right, we call them homologous pairs. Homologous, that's how you pronounce it. Homologous, homologous pair of chromosomes. They're lined up right next to each other. Now, each chromosome is made up of a chromatid. Okay, so that's this right here. This line right here is a chromatid. It has the same information as this guy over here. Well, in enough situations. In meiosis, it's a little bit more complicated than that. They're joined at the centromere, all right? Joined right in the middle. So this point where they're making contact, each sister chromatid, the point of contact is called the centromere, all right? So when referring to a specific pair of chromosomes, we're calling them homologous. So the terminology I expect you to know from this is homologous, centromere, chromatids, and chromosomes. Again, chromosome is the whole operation. Chromatid are the halves that make up the chromosome. So for example, if you're watching these videos in order, you just got done with mitosis. So during that component of anaphase, remember when we divide the chromosomes and pull half to each end, those things that are getting pulled to each end of the cell are chromatids. Okay, because remember, the chromosome is lined up right down the middle, and then this is during metaphase, by the way. They line up right down the middle during metaphase of mitosis. The chromosome lines up right in the middle during metaphase of mitosis. And then the sister chromatids come apart at the centromere and get pulled to opposite ends, okay? So that's why the distinction between chromatid and chromosomes are important. The chromatids are the halves that make up the chromosomes. Now, with that being said, we're going to talk about autosomes, sex chromosomes, what makes a biological male, and a biological female, Okay, so we've already gone over several times by now that humans have, if everything goes well, 46 chromosomes in total with 23 pairs. The 23rd pair is what determines your biological sex. So, for example, if you are a biological female, your 23rd pair of chromosomes consists of an X and an X. If you are a biological male, you have an X and a Y. The reason why we are not able to create another human being with two Y chromosomes, because remember what I just said, a female is two X's and a male is an X and a Y. Notice the similarity between those two. So they both have an X in them, and a human being cannot be made, without exception. You cannot make a human being without at least one X chromosome. The reason for that being is there's not enough information to make anything if you just have a Y chromosome. The X chromosome has so much of the critical information needed to create a human being. That's why some of you may have heard that sort of rumor, like humans are by default female. And there is some truth to that. It's not quite that simple, but you know, it's not the most inaccurate thing to ever be said. So what I want you to take away from that is males are X and Y, females are X and X. The easiest way to remember that is the Y chromosome is, without a Y chromosome, there will not be any programming for the human penis. The Y chromosome is a necessary component for that structure. If a human being just has two X's or even has one X, There are many conditions with that. There will not be any penis developed. The Y chromosome is mostly programming for those secondary sex characteristics and certain hormones. So the Y chromosome is what is programming for the penis, and that is what helps you remember that XY is what programs for a biological male, and an XX is what programs for a biological female. There are certain conditions where they go outside of that. I can talk to you about those if you are curious, but it is not included in the district itinerary for the first semester of biology. So if you're curious, we can talk about Turner syndrome, we can talk about other conditions where it goes outside the boundaries of XX and XY. But for your understanding right now, the XX, female, XY, male. Moving on. So we have 23 pairs. The 23rd pair are our sex chromosomes. The other 22 pairs are called autosomes. Okay, so again, we have 46 total. All of them are in pairs. A pair is 2, so we have 23 pairs. That 23rd pair are our sex chromosomes. The other 22 are just autosomes. They program for everything else. All kinds of fun information. Those of you that plan on learning more about genetics, this becomes very interesting because each number identifies for the same thing on different pairs. For example, on every human being, the 3rd pair programs for the same thing, eye color on every person. It's the same location. It's very interesting to look at, but we don't get quite into that in this class. So let's start on meiosis. Meiosis, like we were speaking about in the previous video, we still have interphase, which has G1, S, and G2. So G1, everything replicates except for the DNA. S, we duplicate the DNA. G2, we make sure everything is Gucci gang before we continue dividing, all right? So interphase is the same in G1, in meiosis and mitosis. I apologize. So interphase is the same in mitosis and meiosis. Now the critical difference is that in meiosis, we have a lot more steps to go through. And we will go through all of those. The easiest way to remember these steps is, remember in mitosis, we had prophase, metaphase, anaphase, telophase. It's very similar. However, we have to do it twice. And these are known in the community as meiosis 1 and meiosis 2. So the way we name each of these phases is we have meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. And then we get into meiosis 2, which has prophase 2, metaphase 2, anaphase 2, and telophase 2. If we were to compare the two, if we were to compare meiosis to mitosis, meiosis 2 we would understand to be more similar to mitosis. But we're going to get into that when we talk about the steps. So by the time it's all said and done, mitosis is going to result in so again, we're talking about mitosis, what we talked about before, not what we're about to talk about. Mitosis results in 2 cells identical to the original. Meiosis, what we're about to talk about, results in 4 cells with half the amount of chromosomes as the original. Because remember, we're trying to make another human being or another living organism. So if I have 46 chromosomes and my partner has 46 chromosomes, and the average human being has 46 chromosomes, we don't need to add our 46 together to make another human being. That's too many chromosomes. That doesn't quantify as a human. So all we need to do to create another living being that's the same species as us is 23 from each. Okay, so I contribute 23, my partner contributes 23, and that results in the 46 that necessitates the creation of another living thing as part of our species, okay? So let's just get right into it. Now, if you don't pay attention to anything else in this video, I want you to know crossing over, and that it only occurs during prophase 1. So crossing over is when one chromosome from your mom and one chromosome from your dad, they literally switch parts of themselves. And I know that can seem confusing, and if you're the type to question everything, you might ask, well, why does that even happen? This ensures genetic variability. We can get more into it in class, but just understand that crossing over during prophase 1 ensures variability. And what I mean by that is, it's because of crossing over that your parents could have a child every day for 1,000 years, and there will never be another one like you. And that is mostly because of crossing over and independent assortment. We'll talk about that later. Without crossing over, your siblings would look a lot similar to you or much like your mom and dad. Crossing over is the reason why sometimes, you'll see a kid and you'll look at the parents and you'll go, what? They don't look anything alike. Crossing over is one of the main reasons for that, okay? They're swapping parts of their information to ensure variability, okay? So what I want you to take away from this is that during the first step of meiosis, prophase 1 is when crossing over occurs, and that is one of the critical components of genetic variability. All right, so just like with mitosis, spindle fibers form, nuclear envelope breaks down. Now, what is different is that they're swapping literal segments of themselves. So your dad, your mom, a segment from each, they just switch components of themselves, all right? Now these two are not the same. These two photos right here, this top one is from mitosis. This bottom one is from meiosis. So metaphase 1 of meiosis, we line up entire chromosomes next to each other, okay, in pairs. Now notice how that's different from mitosis, where we line up each individual chromosome right down the middle, okay? So the critical difference is that in meiosis, what we're talking about right now, we're lining up these chromosomes in pairs. Notice how we got a chromosome here and a chromosome here, versus, this chromosome would just be lined up in the middle if we were doing mitosis. In meiosis, they are lined up in pairs. All right, that's during metaphase 1. All right, this can get confusing. Just, watch the video however many times you need. Look at the flashcards, look at other videos. I will put links to them in the description. The main thing you need to know is that during metaphase 1 of meiosis, the chromosomes are lined up in pairs. They are not lined up directly down the middle like they are during mitosis, all right? They are lined up in pairs, okay? So remember, it follows the same pattern as mitosis in terms of PMAT, prophase, metaphase, anaphase, and telophase. So what do you think happens next if we're still doing PMAT? So we're talking about meiosis. All right, so what is the next step? This is metaphase right here. So it's prophase, metaphase, and what comes after? Just have a little Dora the Explorer moment. Anaphase, very good. So it's during anaphase that we're taking those paired chromosomes and dragging them to opposite ends. So we're taking the entire chromosome and pulling it to opposite ends. Keep in mind that during mitosis, we tore apart sister chromatids. Because remember, let's see if I can, here we go. So remember, during mitosis, we tore apart sister chromatids, those halves, to opposite ends. It's during anaphase 1 that we're pulling apart entire chromosomes, all right? So it's during anaphase 1 that homologous chromosomes are separated, okay? Now we've got telophase 1 of meiosis. So what's different here is that these two are not identical to each other. They are different, and that's okay. We like genetic variation when we're talking about sperm and eggs, because that ensures that our species doesn't put too much stake in one genetic sequence. And we can talk more about that when we get more into evolutionary biology. For right now, I just want you to understand that the cells that result from meiosis are not identical. It's mitosis where we have cells that are identical. All right, so this is telophase 1 of meiosis. All right, so this results in two cells that are different from each other and the original. So we've gone through prophase 1, metaphase 1, anaphase 1, and telophase 1 of meiosis. Remember, meiosis, we go through PMAT twice. There's PMAT 1 and PMAT 2. So we're not done yet. Prophase 2. There is no interphase between the two. Some students get confused and just assume for themselves that there's an interphase between the two. There is not. We go straight from telophase 1 to prophase 2. So for prophase 2, metaphase 2, and anaphase 2, and telophase 2, these steps are very similar to mitosis. Before, during meiosis 1, there were all those homologous chromosomes and all that and crossing over that made it very distinct. During meiosis 2, it's basically the same thing as mitosis. So during prophase 2, chromosomes condense, nucleus breaks down, spindle fibers appear. You know, the thing we're used to. Now metaphase 2, now what makes this different from metaphase 1 is, so take a second to look at this. Look at this image right now in metaphase 2. All right, take a second to absorb that. Notice how the chromosomes are lined up right down the middle. I mean, they may be different at parts, but the chromosomes are lined up right down the middle. Now, look at this bottom part right here. See how that's different? In metaphase 1, they were lined up in pairs right next to each other. Okay? In metaphase 2, they're lined up right down the middle like they were during mitosis. Okay? So that's how metaphase 2 is different. They're lined up directly down the middle, not in pairs. All right? I know this can get very confusing because there are literally 8 steps to meiosis. But just remember, you've got PMAT 1 and PMAT 2. Okay, that's the easiest way to remember it. You got prophase 1, metaphase 1, anaphase 1, and telophase 1. And the same thing for 2. Okay? For this unit, I generally recommend looking at the flashcards first, going through those 2 to 3 times, and then maybe looking at this video. Okay, sort of bringing it all together. So prophase 2, they're lined up right down the middle. Okay, they're not lined up in pairs like they were for metaphase 1. Okay, then we get into anaphase 2. Okay, so recall that during anaphase 1, we were pulling entire chromosomes apart. During anaphase 2, we split up the chromatids that make up the chromosomes. Okay, so it's just like what we were saying during mitosis. But we're in meiosis right now, remember. So again, make that distinction, okay, between the homologous chromosomes and the sister chromatids. And if you're confused, ask me about it during class and I will happily articulate it to you, okay, because this is where a lot of students get tripped up. You've got homologous chromosomes lined up next to each other in pairs. This one, you've got chromatids being pulled apart to opposite ends. All right, that's anaphase 2 of meiosis. During telophase 2, now keeping in mind this is all part of a cycle. We started with 1 cell, now we're ending up with 4. During telophase 2, these nuclear envelopes begin to form around each chromosome. I can almost guarantee you for your test, they are going to ask you how many chromosomes are in each cell at the end of meiosis. And you're going to say half the amount is the original. So this happens in every human being. It's in me, it's in you, it happened in your mom, it happened in your dad. Okay, these cells right here, whether in biological males they're making sperm or biological females they're making eggs, they started with 1 regular cell and they ended up with 4 unique cells different from the original and different from each other that each have half the amount as the original. So you and I, we're human beings, we have 46 chromosomes. Each of these has 23 chromosomes, okay? So if we were the type of organism that had 50 chromosomes, how many would each of these have? If we had 50 total chromosomes, how many would each of these cells have? 25, very good, because half the amount of the original, okay? So again, I know that was a lot. I know it's very complicated, but if you can get through this, it doesn't matter what else is thrown at you for the rest of your high school career, you will get it and understand it, okay? This is probably one of the more complicated things that you guys are going to be exposed to, and I will do everything in my power to make sure you understand it, okay? Just look at the flashcards, watch a video. You can watch it at twice speed, whatever you need to do. I will explain it to you. I am available during first and fourth period. Whatever you need, I'll be there, okay? Just try to understand this, okay? Don't get frustrated if it's difficult. Just come talk to me, ask me where you need clarification, and I will let you know, all right? Don't stress out. Try your best, okay? That's all I ask. Just try your best, okay? I hope you guys have a wonderful day, and catch you next time.